Hello, today we're covering chapter 17 where we talk about gene expression, how we take our DNA and we get that to actually transform into a protein. And we've got this picture right here of these burrows. You can see that some have the protein for coat coloration, so they've got that nice dark coat coloration, whereas others are missing that protein, and so we've got those albinos. And that's what we want to talk about, is how does our DNA actually make a functional protein? And so that's all of chapter 17, or in other words, this is the transcription translation chapter, and at the end we'll talk a little bit about point mutations. Just to remind ourselves about how genetic information works, DNA information is in the sequence of nucleotides. So it means something to a cell when the DNA says AAG versus AAT or AAC. So that sequence of nucleotides is information. DNA, what it does is it leads to specific traits by dictating the synthesis of proteins. This is called gene expression, and gene expression, as we talked way back in the very first week of school, is described as both transcription and translation, which is what this chapter wants to look at. So, basic principles of transcription and translation. Transcription uses DNA to synthesize RNA. So it's this first arrow that we're looking at. DNA makes RNA. That's transcription. Transcription makes a special type of RNA called messenger RNA. Messenger RNA is often abbreviated with a little letter M in front of it. That M stands for messenger. In prokaryotes, this process takes place in the nucleoid region because that's where their DNA is located. In eukaryotes, this process occurs in the nucleus because that's where our DNA is associated. From this point, once we've got the mRNA, that messenger RNA made, then we're going to go through the process of translation. Translation is the process of taking the mRNA and then making a functional protein. Whether we're talking about prokaryotes or eukaryotes, translation always occurs at the same place. It's going to occur at the ribosome. And since all cells have ribosomes, all cells are going to be able to complete this process. This entire process of transcription and translation is what we mean when we talk about gene expression, or what's sometimes also called biology's central dogma. Uniquely in eukaryotes add in a little extra step right in the middle. So eukaryotic mRNA is going to be modified through a step called RNA processing. This is something that we'll look at again today. This is unique to eukaryotes. So big picture wise, before we get into the weeds, as we think about chapter 17, we're learning about gene expression. Transcription first, followed by translation. Transcription is where we go from DNA to messenger RNA. It happens either in the nucleoid or the nucleus. If you're a eukaryote, you then do an RNA processing step. Once you've processed your RNA and you've got that mRNA transcript, you then go to translation, which is this process of taking your mRNA and making the protein at the ribosome. Here's a nice image of this happening within a prokaryotic cell. So you can see the DNA is being transcribed into RNA. Notice that it's not the entire DNA strand that's being transcribed. Instead of making a copy of all of the DNA, we're just transcribing the gene of interest. Maybe this is a gene for an enzyme or a protein required in some metabolic pathway. After doing transcription, immediately that mRNA strand is going to be used for the process of translation. And there you can see there's our ribosome. That's the brown little hamburger bun looking thing. And then what we're making is the polypeptide. These little purple dots, that represents what's going to become our functional protein. So again, this is gene expression in prokaryotes. Transcription followed immediately by translation, making that functional protein. In eukaryotes, the process is slightly different. Notice that when we start doing transcription, that transcription is happening within the nucleus because that's where our DNA is. As we transcribe our DNA, we make an mRNA strand, but then that mRNA strand is going to be processed. This is different than what we saw in prokaryotes. You can see that in the processing, there were some parts cut out. There were also these yellow things added. Again, we'll learn about those steps when we talk about RNA processing. Once the RNA has been processed, it's going to leave the nucleus to get out to the cytoplasm where the ribosomes are. Once it's out in the cytoplasm, it can attach to a ribosome. Again, the ribosome is that hamburger bun looking element, and we start making our protein, which is the purple element. So this is the process of gene expression within eukaryotic cells. It turns out that this process is pretty well understood. There are 20 different amino acids, but only 4 nucleotides. So what scientists had to figure out was, how does DNA tell us which amino acid to build a protein from? It can't be one-to-one. It can't be A means this, T means this, because there's 20 amino acids, 4 nucleotides. It just doesn't work out. In order to make this work, what happens is that DNA is read within a triplet code. So 3 nucleotides at a time will equal 1 amino acid. So AAT will mean something. GGA will mean something. So you read 3 nucleotides of DNA at a time, and then that puts in a single amino acid. mRNA-based triplets are called codons. Notice that these are found in the mRNA molecule, and they're read in the 5' to 3' direction. This is a great image from your textbook that sort of shows you what this process of transcription and translation would look like. So we've got our DNA strand up at the top, that's the blue thing. And what happens in transcription is you're going to read that DNA strand, and then using our base pairing rules, we're going to make the complementary mRNA strand. So we're focusing in on this top strand of DNA, and we're making the complementary mRNA strand. And you can see that A's would match up with T's normally in DNA, but this is mRNA, so we have to put in a U. C matches up with G, C matches up with G, A, that equals U. And you can see that we've now made this mRNA strand. Each of those 3 nucleotides on the mRNA strand is called a codon. At the ribosome, we're able to read those codons, and each single codon codes for the placement of a very specific amino acid. Just as a reminder, amino acids are the monomers of proteins. So if you link lots of amino acids together, that's how you're going to eventually have your protein with its primary, secondary, tertiary, and potentially quaternary structure. What I'd like you to do right now is just see if you can do it. I've got a piece of DNA down here at the bottom of the slide. I just want to see if you can transcribe it. So again, transcription is that first step where you take the DNA and you use your base pairing rules and write down what the mRNA strand would look like. Go ahead and just try it. I'm going to go ahead and start trying to transcribe it. We'll see if I can write small enough. T's will match with A's, C's will match with G's, T's will match with A, so it starts off AGA. Now we've got an A in the DNA. Remember that there's no thymine in RNA, so that adenine is going to need to pair up with a uracil. So that A is going to match up with a U. We'll just keep going. G, C, U, C, G, A, U, U, A, A. That process that we just did down there at the bottom of the screen, that's transcription, where we took a DNA strand and then we built the corresponding mRNA strand. This is called mRNA, messenger RNA, because it's serving as a message to get from the DNA that's encoded in the nucleus out into the cytosol. So what I've done here is I've shown you the genetic code. This is a chart that you do not need to memorize, but you do need to feel comfortable using. It turns out that there are 64 possible different mRNA codons. So 64 different combinations of A's, U's, G's, and C's. And we know what each one of these codons will code for. 61 code for amino acids. 3 are stop signals to end translation, and there's one really important one that you have to memorize called the start codon. It is important that as we look at this, you recognize that AUG is our start codon. When translation starts, it's always going to begin at the codon AUG, which codes for the placement of the amino acid methionine. You should memorize this. So that is really important to just memorize. Of the other codes, you would be provided this type of chart. So if the DNA reads if the mRNA codon reads CUU, we would put down the amino acid LU. You'll notice that there are 3 codons up here in the upper right corner that are called stop codons. These do not code for the placement of any amino acid. What they do is cause the entire translation machinery to stop. The genetic code is a little bit redundant. For instance, CUU means leucine, but CUC also means leucine, right? So there's some redundancy built into the genetic code. However, it is never ambiguous. It's not, well, sometimes CUC means leucine, but other times it means valine. No, it always means leucine. So what I want us to do is practice this a little bit with the DNA strand that you previously transcribed. So on the last slide, you guys took a piece of DNA and you transcribed it into this strand of mRNA down here by this green arrow. What I'd like us to do is take that strand of mRNA and try to translate it using this code that we have shown for us. So again, I'd really like you to try this on your own. Sort of pause the video, look at the mRNA strand, this one down by the green arrow, use this chart that we have shown on the screen, and show me what the resulting protein would look like. All right, I'll try to help out. The first thing that you need to remember is that we are not going to start with AGA. When we start translation, we are always going to look for the start codon AUG. We're going to ignore that first letter A and G. We might have to ignore a whole bunch before we get to that first codon, but we're always going to start with the with the codon AUG, which will code for the placement of the amino acid MET, which stands for methionine. After we've put down that first amino acid, we're then going to continue reading the codons. So our next codon is CUC. If we look over on our chart, you can see there it is. CUC stands for leucine. So our next amino acid is going to be leucine. Our next codon is GAU. If we look for GAU, there it is. ASP. And then our last codon that we've got is UAA, and when we find UAA, we see that it is a stop codon, and so this entire process starts. And so what we've done is we've taken our mRNA strand, this thing that we had transcribed previously, we took that mRNA strand, and we were able to now synthesize this polypeptide, which will turn into the functional protein. You should feel confident doing this. If I give you a DNA strand, you should be able to transcribe it into RNA. If I give you an mRNA strand and this chart, you should be able to use this chart to then translate that mRNA strand. So here's another practice problem for you. Let's see if you can do it. There's an mRNA strand. And I want to know what the second amino acid is going to be. So again, remember the trick is that you have to look for AUG first, and then once you have found that AUG, that start codon, you can then start building the protein. In this case, CUC is going to code for leucine. Leucine would be your second amino acid. The nice thing about this genetic code, and the reason that's so important in biology, is that this genetic code is nearly universal. AUG means methionine, means start, in every living organism that we understand. And so what we can do is we can take DNA from one organism and put it into another organism, and it's still going to be functional. This is how you get things like what we've got on the bottom, where we've got this firefly gene within a tobacco plant. It wasn't just to make a tobacco plant glow in dark at night. We are using this gene to follow a molecule through a metabolic process, and light is easy to follow. Or if we look at this pig over here with sort of the funny coloration on the snout and on the hooves, this pig is having a jellyfish protein, green fluorescent protein expressed. This is not just some sort of Frankenstein-y, weird, mad scientist idea. This is commonly used in biology, and it's a really important tool. If you know somebody with diabetes, they are probably benefiting from our understanding of the fact that the genetic code is nearly universal. If you know somebody who's diabetic who needs to take insulin, what we've done is their bodies aren't able to produce insulin normally. And so what we've done is we've taken a copy of somebody's DNA that does know how to make the insulin protein, and we've cut out that DNA and we've plugged it into a prokaryote, into a bacteria. And that bacteria, when it sees that DNA, it'll just start making insulin. And that allows us to capture that insulin and then package it and provide it to those that have diabetes. Here we have a great review back from chapter 16. Hopefully this is familiar to you. As you look at it, hopefully you should be able to identify some elements. Here we have a replication bubble with origins of replication. Each side of that replication bubble is called a replication fork. We've got our leading strand and our lagging strand. The leading strand is going continuously into the replication fork, whereas the lagging strand is being made as a series of Okazaki fragments heading away from the replication fork. There isn't 5' or 3' labeled on this particular image, but you should be able to identify that right here would be a 3' end, because that's where DNA polymerase 3 is adding new nucleotides. That would make this right here 5', 3', 5', 3', 5', 3', 5', 3'. So again, you should be comfortable with all of these different enzymes. I really like this picture. Please take a moment, see if you can name all of these different colored elements and remember what they do. As a bonus, can you remember what the name of the enzyme that isn't drawn that would be functioning up here upstream is? What is its name? How does it function? So when we think back to what we did in chapter 16, this process of DNA replication, it took a lot of different enzymes to make this work. What we're going to do now in chapter 17 is we're going to start looking at the detailed process of transcription and translation. The nice thing for us when we start looking at the detailed process of transcription and translation is it requires fewer players. So we're not going to need to memorize as many different enzymes and how they function. So let's start with transcription. We're zooming in. We're trying to look at the process of how do we take the DNA and create an mRNA molecule. The enzyme that does transcription is an enzyme called RNA polymerase. This is an amazing enzyme. It does it all. It can pry the DNA molecules apart. It can add new nucleotides to a free 3' end, and it's able to do the entire process of transcription. So its name is RNA polymerase. Please know that name. RNA polymerase can pry the DNA strands apart, add new RNA nucleotides, and it's able to substitute uracil for thymine. In order for RNA polymerase to do transcription, it needs to know where on the DNA molecule to bind. We're not transcribing the entire length of DNA. Instead, we're just focusing on a small portion of the DNA. So in front of every gene, there is a section of DNA called the promoter. The promoter is like a promoter in an event that says, hey, over here, I'm the gene for insulin, or hey, over here, I'm the gene for freckles. So RNA polymerase has to bind to the promoter region. Transcription will then happen. In eukaryotes, it reaches the end of the gene and then just keeps going for a little bit longer and flops off. In prokaryotes, there's a sequence called the terminator. This is a sequence that when the RNA polymerase hits it, it will signal the end of transcription and the entire process ends. Again, that's only in prokaryotes. So we think about transcription. There are 3 stages of transcription. We need to initiate it, we need to elongate it, and to terminate it. And just to keep ourselves making sure that we understand big picture what we're doing, we're talking right now, how do we go from a DNA molecule to create the mRNA molecule? In a prokaryote, this is happening in the nucleoid region. In a eukaryote, this is happening in the nucleus. And the M is used because we are making messenger RNA. This is a messenger molecule that's serving as an intermediary between the DNA and the protein. This is a working copy. So let's look at initiation. In order to initiate transcription, the promoter region, shown in green here, is where the RNA polymerase will bind. So to initiate transcription, we need to have RNA polymerase bind to the promoter. It will then begin to pop open the DNA strand and start adding new RNA nucleotides. That process of the RNA polymerase pulling apart the DNA strands and adding new RNA nucleotides to make this RNA transcript, that's the elongation step. This will continue as this RNA polymerase molecule continues moving down the DNA molecule until we hit termination. In eukaryotes, eventually RNA polymerase just falls off. In prokaryotes, we have the terminator. Let's begin by talking about initiation of transcription. Eukaryotic promoters contain what's called a TATA box. We're not being very creative with our naming. The TATA box is just that, a sequence of letter T's and letter A's, thymines and adenines in that region of DNA. There are proteins called transcription factors that will bind to the promoter, and then RNA polymerase can begin transcription. Here's a great image of this. So we're sort of zoomed into the front of the gene. So here's my gene in the dark blue. In front of the gene in green, that area is the promoter region. And within the promoter region, I have this TATA box. So to initiate transcription, what happens? Transcription factors, those purple blobs, are going to bind to the TATA box in the promoter region. Once the transcription factors have bound to the TATA box in the promoter region, then our enzyme RNA polymerase II is able to bind, and we're able to initiate transcription. After initiating transcription, we then go to elongation. During elongation, RNA polymerase is going to be able to pull apart the DNA strands, and it's going to be adding new RNA nucleotides to the 3' end. Remember, this is a polymerase, so it's always going to need to add to a 3' end. The nice thing about this specific RNA polymerase is that it's able to do this from scratch. That then leads us to termination. In bacteria, RNA polymerase will stop transcribing after hitting the terminator. In eukaryotes, the RNA polymerase will transcribe the gene and then eventually blah, 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 flop off. So that's a nice overview of transcription. Next, we want to talk about RNA processing. Remember, this is something that only eukaryotic cells are able to do. There's 3 elements of RNA processing, and we can see it really well in this picture. So once you're done with transcription in us, in eukaryotes, you've got this long, what we call a pre-mRNA molecule. We are going to make it shorter. You can see how it's a little bit smaller. That's called RNA splicing. We're going to add these 2 yellow bits. One at the 5' end is called the 5' cap, and the other at the 3' end is called the poly A tel. So when we talk about RNA processing, we're talking about those 3 events. RNA splicing, 5' cap, poly A tel. These modifications are really important because if the mRNA does not have these modifications, it's unable to leave the nucleus and get to the cytoplasm. The 5' cap and the poly A tel help protect this mRNA from hydrolytic enzymes. If we think back to viruses, remember viruses infect us with their genetic information and then they take over cells. So cells have a lot of defenses against just random molecules of RNA that are floating in a cell. So what happens is that we need some way of protecting this RNA. And so you can think of this yellow region right here and at the 3' end as being protective. If an enzyme sees this and tries to attack it, doesn't recognize that it's needed, this is going to protect the mRNA. The other importance of these modifications is they help the ribosomes understand which side is the 5' side and which side is the 3' side. Because the 5' cap looks different than the poly A tel, this is a really easy way for the ribosome to know the orientation of this molecule. As we look at this picture, you'll notice that there is, within the red region, some dark red regions and some lighter orangish colored. Not all of the mRNA transcript is coding. Not all of this dark red information tells me how to make a protein. Some of this information is non-coding, and that's what's represented by these light orange regions. So those light areas are genetic information that do not tell me how to build a protein. In eukaryotes, then, what we need to do is something called RNA splicing. Eukaryotic genes have long non-coding stretches of nucleotides that lie between coding regions. So here is one of those non-coding regions. These non-coding regions are called introns. In RNA splicing, what we need to do is we need to remove these introns, right, these non-coding segments, and then we need to glue together the actual coding segments, right, these three dark red parts. So in RNA splicing, what we're going to do is we'll remove the introns, the non-coding elements, and we'll splice together the coding elements called the exons. In order to do RNA splicing, this is sometimes carried out by a molecule called a spliceosome. Spliceosomes are what are drawn down here at the bottom of the screen. So the spliceosome is that big purple and red blob thing with all those smaller blobs in it. That's a spliceosome. A spliceosome is composed of proteins, the purple part, and several small nuclear ribonucleoproteins. I'm going to circle a small nuclear ribonucleoprotein right now. It's that smaller element. So that small circle plus that red piece on the inside. No one ever calls these small nuclear ribonucleoproteins because their acronym is so much funner to say. These are called SNRPs. So a spliceosome is made up of a protein and SNRPs. This particular spliceosome drawn right here has 4 SNRPs in it. A SNRP, what is that SNRP made out of? That SNRP is also made out of a protein and then a special kind of RNA, that's what the red thing is, called small nuclear RNA. And what this spliceosome is able to do, it is able to cut out the intron and then glue together those exons. The spliceosome is an example of something called a ribozyme. Ribozymes are RNA molecules that function as enzymes. And right now you should be like, what? That's crazy. Up until this point, every time we've talked about enzymes, I've mentioned that enzymes are protein catalysts. But here I'm showing you an RNA molecule that has enzymatic function. When ribozymes were first discovered, like a spliceosome, this really changed the biological belief that all enzymes had to be proteins, and we now recognize that enzymes are often proteins, but things such as RNA and other nucleic acids can function as enzymes in some situations. Introns are important. Please don't think of them as just junk information that they're cutting apart. They often contain sequences that may regulate gene expression. So this might be a promoter, this might be a place that a transcription factor binds. So even though they're not coding, they still are important. Introns are also important because of a thing called alternative RNA splicing. What happens, especially in eukaryotes, especially in our cells, the number of different proteins that I can make is much greater than my number of genes. It turns out that human beings do not have very many genes, especially when compared to many plants. What happens is we take our few number of genes and we just cut them and paste them together in different ways to create different protein products. Here you see a great example of this. We've got one gene here in blue. You can see it has a different variety of introns and exons. If we splice together those introns in one fashion, we may make one protein. But if we splice together those exons in a slightly different way, we get a second protein. So we might just have one gene, but by splicing those exons together in different ways, we can create multiple protein products. This gets us to the end, sort of in that middle step. So we've been through transcription. We saw initiation, elongation, termination. We learned about RNA polymerase. We then just looked at this RNA processing step that happens only in eukaryotes, and we're about to move on to translation. Translation is where we're going to actually take the mRNA strand and build the polypeptide. In order to do translation, we're going to need to use a new molecule. This molecule is called transfer RNA. It's often abbreviated tRNA. tRNA molecules are molecules that are transferring amino acids to the ribosome to build the polypeptide chain. This little green thing right here is how our textbook likes to draw these tRNA molecules. Another thing that we'll need in order to do translation is the ribosome. Again, it's this big brown hamburger bun looking molecule. The ribosome is where translation will take place in both prokaryotes and eukaryotes. Molecules of tRNA are not identical. A tRNA molecule is a short RNA strand that has been folded back on itself. Each tRNA molecule will have a very specific amino acid on one side. So up here, there'll be an amino acid that gets attached. Maybe that's methionine or tryptophan or valine. And then on the bottom side, they're going to have an anticodon. The name already gives it away, but the anticodon will base pair with the complementary codon on mRNA. And so although this is letter B, visually what they look like, to simplify the tRNA molecule, you're often going to see this symbol. Accurate translation, then, is going to really require two important processes. Number one, I've got to make sure that the correct amino acid is on the correct tRNA molecule. So there has to be a match between all these different tRNA molecules and all these different amino acids, and I've got to make sure that the right ones pair up. In order to do that, I'm going to use an enzyme called aminoacyl tRNA synthetase. Yes, you do need to know that long name. The second part of translation is that I've got to make sure that the right codons from mRNA are pairing up with the right anticodons from the tRNA. This is going to be the role of the ribosome. So two important steps in order to have translation occur appropriately. Let's start with the first one. How do we make sure that out of all of those different tRNAs and all of those different amino acids, we get the correct ones pairing up? Let's think it through a little bit. Let's imagine that I've got a tRNA anticodon that reads AUA. What codon would this anticodon pair up with? Some things to remember. The codon is also an RNA molecule, right? That's our mRNA molecule. So if my tRNA reads AUA, right, that's the tRNA molecule, we can just use our base pairing rules to figure out what the mRNA molecule would read. So A's would match up with U's because this is an RNA molecule, AU. So the answer to this second question, or that middle statement, what codon does the anticodon pair up with? The answer would be it pairs up with the anticodon UAU. Okay, next question. Down here with sort of my red arrow. What amino acid would the codon UAU place? We can use our codon chart. Again, you don't have to memorize this. So let's look for UAU. Here it is up here at the top. The mRNA codon UAU means that we're going to put down the amino acid tier or tyrosine. Okay, so I know now that if I've got a tRNA molecule that has the anticodon on it AUA, right, that it should match up with an mRNA molecule that reads UAU, and it should be able to pick up the amino acid tyrosine. So what are we going to do? There is an enzyme called aminoacyl tRNA synthetase that does that matching for us. That work that we just did on the previous slide, it's able to do. And so what it does is it picks up a tyrosine amino acid. It also picks up that tRNA molecule that has that anticodon that we were just talking about. There's the anticodon, AUA, just like we had on the previous page. This enzyme brings those pieces together. It then adds energy, and it is able to attach the correct amino acid to the correct tRNA molecule. And again, remember, this is our first really important step to happen in translation. We've got to make sure that there's the correct amino acid on the tRNA molecule. And this enzyme, aminoacyl tRNA synthetase, is doing that for us. Next, we need to make sure that this tRNA molecule actually gets and matches with the mRNA molecule. To do this, we're going to use a ribosome. The ribosome is where we match tRNA anticodons with mRNA codons. There are two subunits, a large subunit and a small subunit. These are made up of both proteins and ribosomal RNA. So when you look at this big brown blob, it's not made out of a phospholipid bilayer. This isn't a membranous organelle. This is a protein plus a bunch of RNA called ribosomal RNA. So there's another type of RNA that you should know about. rRNA stands for ribosomal RNA. It's the component of the ribosome. It's what we build the ribosome out of. As you look at the ribosome, you can see that there are 3 different sites, an E site, a P site, and an A site. They always are going to be in this order, so you can think EPA, like Environmental Protection Agency, and that might help you remember them. Specific events will occur at each of these phases. You need to know what occurs in each site of the ribosome. Let's start from the right and move towards the left. In the A site, what happens is a tRNA molecule will come in holding A amino acid. So you can use that A to help you remember what happens. A tRNA molecule comes in that's holding A amino acid. In the P site, here in the center, you have a tRNA molecule holding on to a polypeptide chain. So use that P to help you remember what's going on here. Polypeptide. The tRNA molecule is holding on to that long growing protein chain, that polypeptide chain. The E site is where the empty tRNA molecules will exit. So here you can see this tRNA molecule. He's empty. He doesn't have any amino acid at the top, and so he's able to exit the ribosome. Please know specifically what the tRNA molecule is doing at each site in the ribosome. Now that we have all these players together, we can look at the 3 stages of translation. The 3 stages of translation are initiation, elongation, termination. Just as a big picture review, remember in translation what we're doing is we're taking the mRNA molecule and we're trying to build that functional protein, and this is happening in the ribosome. So to initiate translation, we take a small ribosomal subunit, and it's going to begin by binding with the mRNA molecule. So this down here is the very first thing that happens. Small ribosomal subunit, mRNA lands on top. After that, the initiator tRNA will come in and bind the start codon. So you can see that that initiator tRNA is looking for the start codon. So that's step 2. Step 3, the large ribosomal subunit will come in and bind. When that large ribosomal subunit slides into place, it's going to make sure that that initiator tRNA molecule is in the P site. From here we go to elongation. A tRNA molecule holding a single amino acid will bind to the A site. This is where the codon will pair up with the anticodon. Peptide bond will be formed. An amino acid in the P site will bind to the amino acid in the A site. Then there will be a translocation. It's a fancy word to just say the entire molecule will shift. The E site, there will now be an empty tRNA that can exit. In the P site, there will be a tRNA molecule holding a polypeptide chain. And in the A site, there will be a tRNA molecule holding a single amino acid. Your textbook has a great picture of this elongation phase that we'll work our way through. So here we go. Here's our mRNA strand in red, the ribosome in brown, and in the P site, we have a tRNA molecule holding a polypeptide. So in order to do elongation, first thing that happens is a new tRNA molecule holding A amino acid will come into the A site and bond. So this is where the codon is pairing up with the anticodon. Once that has occurred, we're going to make a bond. We're going to make a peptide bond. We're going to take this long polypeptide chain, and that polypeptide chain that was in the P site is going to bind to the amino acid that is in the A site. We just made that bond. You can note now in this bottom picture that everything seems to be in the wrong spot, right? My empty tRNA is in the P site. My tRNA molecule holding the polypeptide is in the A site. None of this makes sense. So the entire ribosome is going to translocate. It's going to shift. Here we can see that shifting. Now we have our empty tRNA that can exit. Our tRNA molecule holding that long polypeptide chain is in the P site, and now the A site is open and available for the next tRNA molecule to come in. And this process is going to repeat over and over and over and over again during elongation. This occurs until we hit termination. In termination, there is going to be a stop codon in the mRNA that reaches the A site of the ribosome. When that happens, the A site will accept a release factor. Down here at the bottom, you can see it. It's that weird yellow-looking molecule. The release factor causes the addition of a water molecule, good old hydrolysis, way back from chapter 5. When water is added, this causes the entire machinery to come apart, right? We're going to release the polypeptide. All these pieces are going to fly away. Out of all of these pieces, it's this thing, that polypeptide, that I'm interested in. That's the whole purpose of this process. I've been trying to make this polypeptide, this protein, to take that original DNA, to transcribe the RNA, to then modify it if I'm a eukaryote, and finally end up with this polypeptide. This process is a very visual process. I really encourage you to go into Mastering Biology and watch the animations and the videos associated with transcription and translation. They'll do a much better job than I can do with still pictures showing you this process. Within the study area, there are some 3D animations. There are also figure walkthroughs. I would encourage you to watch all of those for the related transcription and translation elements. Please, please, please use those resources. I think it'll really help you tie these processes of transcription, RNA processing, and translation together. Now that we have our polypeptide chain made, these are often going to be modified after translation. We're going to fold them, we're going to stick sugars on them, that's called glycosylation. We also have to transport them, maybe using our endomembrane system, to the appropriate locations within a cell. In order to get a protein to the appropriate location, it happens very early on in the process of translation. Remember, we have two different types of ribosomes. One ribosome is called a free ribosome. Those are the ones that are just floating around the cytosol. And then we have bound ribosomes that are stuck on the surface of our rough ER. If you are a free ribosome, you're floating around in the cytoplasm, you are going to make proteins that are also going to be floating around in the cytosol. If you are a bound ribosome, you are going to make proteins that are going to be embedded in membranes or that might be secreted from the cell. Polypeptide synthesis always begins in the cytosol. So when I start making a protein, when I start doing translation, I always start on a free-floating ribosome. If this polypeptide, if this protein is a molecule that needs to be embedded in a membrane or need to be secreted, it's going to be marked at the very beginning by the sequence of amino acids called a signal peptide. It's that little green dot. What happens if there's a signal peptide, this will be recognized by another molecule called the signal recognition particle, SRP, that orange thing. The signal recognition particle will bind to the signal peptide, like we see sort of in step 2, and that will draw the entire ribosome polypeptide machinery over to the ER, and will take that free ribosome and change it into a bound ribosome. Polypeptide synthesis is continuing, but now you can see that this protein is going to be synthesized inside the ER, so it can be packaged into membranes or it can be packaged for secretion. RNA, you should be familiar with these 3 types of RNA. M stands for messenger RNA. It's a message molecule between our master copy in the DNA and the protein. It's that intermediary that's able to move between the nucleus and the cytosol. tRNA is your transfer RNA. This is the molecule that's transferring amino acids to the polypeptide. And then ribosomal RNA, this is the actual functional structural component of the ribosome. Multiple ribosomes can translate a single RNA molecule simultaneously, creating this structure called a polyribosome or a polysome. This is a way to increase efficiency within the cell. So as soon as there's room on that mRNA strand for another ribosome to catch on, you can get another one, and this is again going to increase efficiency. In conclusion, we want to look briefly at mutations. We're going to look at a very specific type of mutation called a point mutation. So a mutation is defined as any change in the genetic material. Point mutations are changes where just one letter, one base pair is being changed. We've seen this before when we looked at sickle cell anemia and we looked at that what happened. Hopefully in your normal DNA, this is what the DNA reads. Everything's fine in the mRNA and the protein. In sickle cell anemia, what happens is one base pair, one letter gets changed, so your mRNA is changed, and so you get one amino acid in that protein that's the wrong amino acid, and that causes all the symptoms associated with sickle cell disease. There are different types of small-scale mutations. Within point mutations at the DNA level, we can differentiate between nucleotide pair substitutions and nucleotide pair insertions or deletions. It's really nice. The vocabulary makes sense. They are exactly what they meant. Imagine that this sentence is a structure of DNA. So I know it's not DNA, but let's just imagine the cat was fat. Right? And now what I've done is underneath that original DNA sequence, I've given you some mutations, right? I've given you some point mutations. The cat with the letter K was fat, right? That's a substitution, right? Where I've substituted the letter K in for the letter C. Or this next line, the bat was fat. That's another nucleotide pair substitution. This one right here, the kut was fat, that was really messy. Notice that's what happening is I'm missing the letter A, right? It's missing. And so this is an example of a nucleotide pair deletion. Or if we go to the very bottom one, Z was fat, right? This one, we have an added letter Z in the middle of our DNA. And so this would be a nucleotide insertion. So again, point mutations, or we're just changing one letter in the DNA. If you substitute, you're sort of swapping in a different letter. And an insertion or deletion is either inserting in a whole other letter or deleting out an entire letter. Point mutations in our DNA can lead to then mutations in our proteins. So now I want to focus about the protein mutations. Silent mutations are mutations that have no effect on the amino acid. So yes, the DNA had a mutation, but luckily for us, it didn't cause any change to the amino acid, so the protein is still functional. This happens often because that DNA code, the genetic code, is redundant. Missense mutations, there's a mutation in the DNA which causes the wrong amino acid to be placed in the protein. Think about something like we would see in sickle cell anemia. Nonsense mutations, there is a mutation in the DNA that causes an amino acid change, but the codon change is to introduce a premature stop codon. So what happens is the protein is stopped, the process of translation, early. Even though the rest of the genetic information might be correct, that premature stop codon stops it. The way that I remember this one is, I think no, right? Nonsense has the word no in it. No, stop making the protein. Inserting or deleting nucleotides may alter the reading frame. This creates something known as a frame shift mutation. So down at the bottom, I've got those sentences again. This time I want you to think of these sentences as proteins. So we're looking at a protein, right? Here's our original protein, the cat was fat. And now we've got this protein being mutated in various ways. In our first example, this one with the green arrow next to it, the cat was fat. Yes, there's a mutation in there, right? But the sentence still makes sense, right? The cat was fat. You can't tell which sentence I just read. So this would be a silent mutation. Something like we see with the bat is fat. We've put in the wrong amino acid, right? The letter B is the wrong amino acid. So this would be a missense. This next one, right? Just the TH and then it stops. And there's nothing else. This would be an example of a nonsense mutation. And then at the bottom, we've got some frame shift mutations, right? Our fun little phrases, or right? In both of these cases, we have shifted the reading frame. So the information is still there, but it's been slid out of alignment. And that causes major problems for the organism. Your textbook ends with a beautiful diagram showing all of this. So at the top we have gene expression, the way that it's supposed to work. You've got your DNA being transcribed into RNA, RNA making a protein, and then they go ahead and go through a variety of examples of nucleotide pair substitutions, insertions, and deletions, and how this may affect the resulting protein. This brings us to the end of chapter 17. You should have a good feel now for the process of gene expression, transcription, RNA processing in eukaryotes, translation, and then this brief little look at point mutations. If you've got any questions, as always, please reach out. I would love to answer any questions as they arise.